All right, everyone, we're going to go ahead and get started. Thank you so much for being here today for our seminar. I really appreciate you coming in. If you're not here in Newport right now and you're online, it is sunny outside, which is a great little thing. So thank you, everybody, for being here. My name is Cinnamon Moffett. I'm the Research Program Manager here at Oregon State University's Hatfield Marine Science Center in Newport, Oregon. This is a hybrid event, so we have folks online and folks in the room. A couple of quick instructions for those folks that are online. Your cameras, mics, and screen shares are disabled for this event, but please use the chat box to communicate with us. Our volunteer, Roseanne, is in the room. She'll be monitoring that chat box if you have any technical issues or if you have any questions for today's speaker. For folks in the room, I'm looking around, most of you know this already, but if you have a question for the speaker, just raise your hand at the end of our question and answer section, and I'll bring you a mic, and that way folks online can hear your questions as well. But again, thank you so much for being here. I also just want to do a quick announcement for next week's seminar. Next week's seminar is an invited speaker by our students. So this is our annual event where our students have an opportunity to invite somebody that they think will impact their careers moving forward. So next week's seminar is Leslie Noon. She is an assistant professor of statistics at I'm going to say this wrong, Lisa. Ursinus College, and we'll be talking about statistics, the dark side of ecology. So we're very excited about this particular speaker and the topic that she'll be presenting to us. But for today, the speaker we have here was invited by one of our own postdocs that works up with the Marine Mammal Institute, Dawn Barlow. So I'm going to actually hand the mic off to Dawn to introduce today's speaker.
Thanks, Cinnamon. Thanks for coming and joining us today. And it's my pleasure to introduce today's Hatfield seminar speaker, Will Oestreich. So Will is a postdoctoral fellow at the Monterey Bay Aquarium Research Institute. And prior to his time at MBARI, he got his PhD in biology at Stanford University and completed both his master's and bachelor's in environmental engineering at Northwestern University. He also conducted oceanographic research at Woods Hole Oceanographic Institution and did some policy work with the National Resources Defense Council. And Will's research explores animal behavior in dynamic and changing ocean ecosystems. And I've had the pleasure of getting to know and work with Will over the last several years now, and I'm always impressed by his sort of big picture ecological thinking and creative analysis approaches. So really excited to hear his latest and greatest here with you all today. Take it away, Will.
All right. Thank you so much, Dawn and Cinnamon, and to all of you for welcoming me here. This has been just such an exciting day. I've never been to Hatfield Marine Science Center before, and I've wanted to come here for a long time. So this has been a real dream come true to come here, and to meet and speak with many of you has been a real pleasure. I just really appreciate how welcoming and interested and excited everyone's been in the conversations I've had today. It's really been a warm welcome. I'm really excited to also share some work that I've been doing along with a team of collaborators that you'll see throughout the talk that make this work possible. And yeah, as Dawn mentioned, my interests are really at the interface of behavioral ecology and oceanography. And when I think about those two topics, I really like to start by thinking about, let's make sure this is going to work. There we go. Okay, thank you. All right, first hiccup is behind us. Yeah, so I like to start when thinking about behavioral ecology in this oceanic context. I like to start with these krill because I think they're a really nice representative of what life is like for animals inhabiting oceanic ecosystems. So these animals are amongst the most abundant animals on the planet, both by numbers of individuals and total biomass. And they live in this really interesting existence where they're heavily influenced by both the physics of the ocean, sort of living a somewhat planktonic lifestyle, but they also have the opportunity to exhibit agency through behavior in controlling their distribution and interactions in the environment to some degree. And these physical and behavioral interactions are really central to another central tenet of life in pelagic ecosystems, which is extraordinary patchiness. So even though we have this incredible number of animals and biomass of animals, they tend to be very unevenly distributed in the environment, aggregating in these patches of really dense organisms aggregating together. And these aggregations occur over nested spatial and temporal scales. So we see this patchy nature really even within a group of organisms, across groups of organisms, and then even out to the scale of oceanographic features. The other really central tenet here is that these patches are constantly being moved in space and time, right? This is an extremely dynamic ecosystem, not surprising to a room full of folks like you all that study the open ocean or marine ecosystems more broadly, but this really is an important factor to consider of how different this can be relative to the dynamics we see on land, for example, where things are definitely changing on perhaps longer timescales, but this patchiness is not being distributed in space and time quite as dynamically as we see in oceanic ecosystems. Of course, these animals also have to deal with this other enormous part of life in the ocean, this enormous third vertical dimension, which introduces a really strong gradient of light into their lives. And this whole arrangement of the aquatic medium, the vast scale of these ecosystems, the three-dimensional enormity, the fluid medium, really can select for different behavioral strategies in the predators that inhabit these ecosystems. So we see a variety of animals, for example, searching for these prey patches, using sound for different purposes in the aquatic medium in which they live their lives. So just to recap, if we think about life and behavior in the ocean versus on land, we can think about there's really dramatic differences in ecosystem scale and structure, spatiotemporal patch dynamics, the sensory modalities and communication range that are selected for in these different resource environments. And as someone who really is interested in the behavior of animals and particularly of animal groups, I'm really fascinated by these differences across marine and terrestrial systems because there's a really rich growing body of theory in behavioral ecology about how spatiotemporal ecosystem dynamics and perception, sensing, and communication really interact with collective behavioral processes to drive the interactions between individuals and the evolution of collective and social behaviors in animal groups. So we have, for example, everything from how optimal foraging theory can be altered by spatiotemporal resource dynamics, to how different group sizes and perceptual and communication ranges can be selected for under different resource dynamic scenarios, up to how these social interactions and sensing and communication drives behavior at the collective group level. I'll just pull out one example from here. This was a paper that I was lucky enough to work on with an outstanding undergraduate researcher at Kanoi a couple of years ago. And I won't go into deep detail about this review paper that we worked on together about social elements of animal migrations. But one thing I will point out about this paper is when we look for case studies describing how animals use social information to time their migrations, what we found is this is widespread across the animal kingdom, but almost all of the case studies we could find were in terrestrial organisms. So if we return back to our differences between marine and terrestrial systems here, I think there's a really critical fourth element to consider here. And it's that in one of these, we often have detailed observations of behavior at both the individual and group levels. Whereas in the oceanic system, while we increasingly have these observations, they have been lacking to date. And this is, I think, really important space to study these dynamics given the differences in ecosystem dynamics relative to terrestrial systems. Of course, don't have to convince anyone in this room of this, I'm sure, but this is also important because we're living on an ocean planet, right? This is the vast majority of the habitable space on our planet is this type of ecosystem where these dynamics are at play. And we really don't have a solid understanding of how the physical and biological ecosystem dynamics interact with behavior to drive life in the ocean. Of course, elephant in the room, these ecosystems are also rapidly changing. We know that both anthropogenic climate change and more direct industrialization of oceanic ecosystems is changing the way that these ecosystems function and how animals live in these ecosystems. And given that animals first responses to environmental change is typically behavioral, I think we have a really strong imperative to understand behavior of animals in relation to ecosystem dynamics in order to more mechanistically predict how they might respond to these changes in the near and medium-term future. So my research program, as Don mentioned in the introduction, is really focused on understanding animal behavior in dynamic and changing oceanic ecosystems. And I like to think about each element of this process, from the biophysical ecosystem dynamics, how that interacts with prey behavior and distribution, how prey behavior and distribution interact with predator behavior distribution, and how that feeds back onto the prey. And then the two sort of lenses that a lot of my research is really situated in, looking at this continuation of ideas, is thinking about collective behavioral processes, these interactions between individual and group behavioral dynamics, and then how this is nested under understanding anthropogenic ecosystem change and the role of behavior in the lives of animals in responding to that change. So I'll be talking about a few different vignettes of research today. The first is really focused on understanding drivers and adaptability of collective behavior under ecosystem variation and change, specifically for blue whale predators and their krill prey in the Northeast Pacific. So at the broadest level, just to situate everyone to the population we're talking about here in the annual cycle of these animals, here we're looking at the west coast of North America. The ecosystem, the part of this ecosystem that I really study these animals most intensively is in Monterey Bay and the Central California Current. And Monterey Bay, of course, is well known for its large submarine canyon. It's also a real foraging hotspot for blue whales during the summer and fall foraging months. Let's see if this is going to work for me. There we go. And during these summer and fall foraging months, the availability of krill prey that these blue whales are obligate foragers on is really driven largely by seasonal upwelling dynamics. So of course, just as a quick review, this upwelling is the process by which surface waters are being moved offshore by northwesterly winds interacting with the rotation of the Earth, where it's replaced then by these deep, cool, nutrient-rich waters, which drive an explosion of life across trophic levels, including these dense swarms of krill that blue whales are searching for during their summer and fall foraging season in the California Current. This seasonal upwelling process is seasonal in nature, but that seasonality also varies interannually in terms of its timing and intensity. So we can think about this variability in a few different ways in terms of the total accumulation of nitrate flux as a result of upwelling, the timing of that nitrate flux. And so a question that I've been really interested in is how blue whales handle this interannual variability in deciding when to be foraging and when to be migrating south to their wintering grounds off Central America. So after these summer and fall foraging months, as I mentioned, blue whales are migrating southward to areas off the west coast of Central America to their calving and breeding grounds. And again, the question that I was really interested in thinking about here is, how do blue whales actually make a decision of when to forage and when to migrate in such a vast and dynamic oceanic system that they're inhabiting? And this is work that was primarily conducted during my PhD years and funded by a variety of organizations as shown here on the slide. The main tool that I've been using with collaborators to study this migratory behavior is long-term passive acoustic monitoring. So at the Monterey Bay Aquarium Research Institute, John Ryan leads a long-term passive acoustic monitoring program on MBARI's cabled seafloor observatory, which is shown with the white dot there, just outside of the Monterey Canyon. And this observatory is cabled to the lab on shore, which enables a constant power supply to the instrumentation and also a constant data feed back to the lab in the other direction. We've been recording since the summer of 2015 and ongoing. This generates enormous amounts of data. I'm very lucky to work closely with software engineers that enable us to make this data publicly available, and also with open source analysis tools in Python that allow others to interact with this data, not just as raw data, but also as derived products relevant to animal behavior. And over this really long-term recording period, we've recorded blue whales for more than 1,000 days over what we estimate to be a roughly 10,000 square kilometer detection range based on acoustic propagation modeling efforts with collaborators at the Naval Postgraduate School. I'll just note before I move on here that In case folks are not aware, this acoustic data stream is actually listenable online through the Monterey Bay Aquarium Research Institute's website. So if you're interested to tune into the sounds of Monterey Bay at any time, I encourage you to check it out on the MBARI website. So I've mentioned out blue whale song quite a bit now. What am I talking about when we talk about blue whale song? I'll let the blue whales speak for themselves a little bit here, speeding up the audio playback here to bring it more into our human hearing range. So you'll notice that there's this repetitive pattern that this blue whale is producing, and these songs comprised of different units, which are labeled at the top of the spectrogram here, which is representing sound and frequency and time space here. And you'll notice that the B call in particular is a really strong high amplitude signal in these calls. We take advantage of the very stereotyped nature of that call type to develop a really simple calculation to quantify the abundance of blue whale song in the soundscape at any given time, simply by quantifying the amount of acoustic energy in a target frequency band relative to nearby background frequency bands, which we refer to as a call index, which again is really a proxy for the abundance of song in the soundscape around our recording device. So I mentioned this roughly 10,000 square kilometer detection range around our hydrophone. Here you can see our acoustic propagation modeling result efforts. The areas shown with colors around the circle here are the areas over which we estimate we can hear blue whale B calls above typical ambient background noise conditions, which gives us really nice coverage in space. And again, that really nice long-term coverage in time has allowed us to look at a variety of factors, including the average annual cycle and when we're hearing song at this hydrophone. So that's what's being shown here is many years of recording collapsed down into an average annual cycle of that call index metric. We can also think about these patterns not only in seasonality, but also the interaction of seasonality with diel or daily patterns in song. So what's being shown on here, on the bottom here is the night to day ratio of that call index, with values above 1 indicating more song occurring during the nighttime, and values below 1 indicating more song during the day. And to my eye, there's really two overlapping parts of the season here. There's this first part of the seasonality during the summer and fall months, where song intensity is rising, and the majority of the song is being produced during the nighttime. And then there's this period at the peak of song and then tapering off into the winter months, where not only is song intensity going down, but the proportion of song that's occurring during the nighttime is going down. And I remember the first time that my collaborators and I looked at this plot, we were thinking there must be other elements of behavior that can explain such a strong pattern over such a large spatial scale for many animals over many years of recording. So to investigate what might be driving this population level pattern in song, we turn to individual level methods to understand the individual level behavioral context for those patterns in song. And this is work done during my PhD in Jeremy Goldbogen's lab, where we used biologging tags designed in partnership with customized animal tracking solutions, which we can deploy on these animals to really get a detailed view into the lives of these animals beneath the surface of the ocean, where we are otherwise unable to observe their behaviors in detail. So to give you a taste of the amazing view into the underwater lives of these animals that we get from these tags, here we're watching a pair of blue whales foraging on a krill swarm together, one blue whale filming another blue whale from its back. You can see that amazing lunge feeding behavior that this animal is engaging in at depth in Monterey Bay. And while the videos are lovely and give us an incredible insight into the lives of these animals, the data that comes off of the variety of sensors on these tags give us a really, really rich perspective into the lives of these animals. So an example of this here, this is one tag deployment on a blue whale in Monterey Bay. We can see the dive profile of this animal in black here, with all of the blue circles indicating each of those feeding lunges, which we've identified from the tag data, and then the green circles indicating the singing behavior, also identified from the tag sensors. And you'll notice that this animal spends most of the daytime feeding at depth and most of the nighttime near the surface producing song. This is an example from one individual, but we can also look at many individuals over several years, look at their song call rates, again, seeing this consistent pattern of mostly singing during the nighttime and mostly feeding during the daytime. All of the data that I'm showing on this slide is just data from animals that are in a foraging behavioral state. But we can also think about what animals' behavior looks like from these tags once they initiate that southward migration. So to do this, we use slightly longer duration tags. So here we're looking at a deployment of a tag that stayed on a blue whale for a month. You can see in the inset map here the track that the animal swam roughly from the San Francisco Bay Area down to Baja California in Mexico. You can see the latitude of this animal through time in the black line on the main plot. This dashed line here is indicating the period during which before the dashed line, the animal is in this foraging mode, and then you can see the latitude going down as this animal is in its post-foraging southward migration. Using those tag sensors, we can overlay the more fine-scale behavior of this animal on this tag track. So here you can see the circles indicating how much this animal is feeding each day, the triangles indicating how much this animal is singing each day. We can also look at the time of day that these behaviors were occurring. So if we now overlay some color onto this plot, the red colors are indicating a daytime bias in the behavior, and the blue colors are indicating a nighttime bias in behavior. And we see during this foraging period, again, that familiar pattern, this animal is singing mostly during the nighttime. But once this animal starts migrating southward, we see a mix of blues and whites and reds. This animal has its song spread more evenly over the day and night over this period of weeks. So this is, again, one example. We've seen this for a number of individuals. And it allows us to go back to our population level patterns of song and understand that what we're actually hearing here is a population level shift in behavior. This transition where we're hearing most of the song during the nighttime, dropping off into a more even distribution of song throughout the night and the day, is this population shifting from foraging to southward migration. And this is a really exciting discovery, I think, for a number of reasons. But the first question that came to mind was, if we can now determine when this population is initiating their southward migration, how flexible are they to interact with that interannual variability in foraging conditions that I described earlier in the talk? So is there interannual flexibility in this population level timing of migration? So now the plots I'll show are not the average annual cycle of song metrics, but broken down year by year. So here you can see this, again, this call index or song intensity night to day ratio year by year, where we know the transition to migration is indicated by the drop off in that night to day ratio. So these periods are shown in the red dots here, where we're getting that transition to migration. And we see years like 2015, for example, where these animals migrated late in the summer, early in the fall from Monterey Bay. A year like 2020, where we didn't get a really strong drop off until late in the year. And even a year like 2017, where we saw multiple waves of what we think is this behavioral transition from foraging to migration. So the answer to this question is yes, there's incredible flexibility here, spanning roughly 4 months of variation, which is quite a bit of variation when we're talking about a 12-month annual cycle. The follow-up question there is, are these variations in blue whales foraging and migration behaviors and the timing of those behaviors linked to the variation in ecosystem phenology, that upwelling process that we know drives the availability of their krill prey? I won't get into this analysis in detail here. Happy to take more questions about it if time allows, but I'll just jump to the conclusion here that yes, using a variety of metrics that quantify the phenology of that upwelling process, we found that the blue whales were migrating southward from Monterey Bay later in years with an earlier onset, a later peak, and greater total seasonal accumulation of this upwelling. So if we zoom back out to the annual cycle that these animals are operating in, what we've shown here is this interannual flexibility in migration from the foraging habitat towards the lower latitude habitat. And interestingly, I will note that this is the exact opposite of what's been shown when these animals are migrating in the opposite direction. So work by my collaborator, Brianna Abrams, several years ago used long-term tag data to show that these animals are actually very consistent in the timing of arrival at different latitudes on their northward migration, which Brianna and colleagues attributed to spatial memory to track long-term average conditions. And I think this is really interesting in thinking about the sources of information that these animals are using in different parts of their lives. To me, this makes a lot of sense. When these animals are coming to the foraging grounds, they're coming from a different oceanographic system that may not have information for them to go on when making a decision of when to show up on the foraging ground. So it might make sense to play the long game and hedge your bets by tracking long-term averages. But when these animals are leaving the foraging habitat, they have much more information at their disposal, both their own experience of the foraging conditions they've seen, and also potential information from other whales and their foraging success. So they have a lot more information to go on to making a more flexible decision year to year of when to migrate southward. I'll just note that I've noted that this could be individual and or social cues that could drive this flexibility, which we really haven't teased apart in the work I've shown so far. And this is a follow-up question that I've been trying to think about with collaborators to understand what are the sources of information that actually enable this incredible flexibility that we see in such a vast and dynamic foraging habitat. And this is work that I've been collaborating on with Brianna, who I mentioned a moment ago, and also with my collaborator, Stephanie Dodson, who's with the math department at Colby College. And this is work that's currently in review, but what we've been working on is developing individual-based models or simulations of animal movement in which we either allow blue whales to listen to one another's behavioral state, which is indicated by when they're singing during the daytime or the nighttime, versus simulations where you don't allow the animals to produce or receive acoustic information in your computer world. And using these simulations, we've been able to overlaid on oceanographic data and metrics of krill availability through space and time in the California current. We've been exploring what migration timing looks like when animals use personal information, so that information they're acquiring just from their own foraging experience, versus information that's both personal and social, so listening to others. And what we're finding is that these animals are able to stay in the ecosystem and successfully find krill much later into the year whenever they're eavesdropping on one another's behavioral state. What we're finding is that this is a better match for the empirical observations we make at the population level using hydrophone data, and then also comparing to previously published data by Ladd Irvin and colleagues based out of Oregon State. At the individual level, we find that these animals are departing the US exclusive economic zone in our personal and social model in a way that's a better match with individual level tag data, again, when we're allowing these animals to use social information from conspecifics in our simulations. Just in terms of what that means in terms of krill intake as well, here you can see how much krill these animals are acquiring relative to a hypothetical non-migratory population, which is that line at 100% here. And so we see when we allow these animals to listen to one another's behavioral state, they have much higher foraging success rates at the population level throughout the foraging season. So this is a lot of information that I just threw at everyone about blue whale migration. So just to take a breather here for a second and recap what I'm trying to communicate here. First, using a combination of tag data and long-term passive acoustic monitoring data, we are able to show that the time of day that blue whales are singing can be indicative of their behavioral state. And then using that information and applying it to interannual data from passive acoustic monitoring, we're able to show that this population level migration timing is variable to track interannual variability in the seasonality of their foraging ecosystem. And then finally, using these simulations, we are showing that blue whales potentially could be using this information on conspecifics behavioral state to make better informed decisions about when to give up on the foraging season and migrate south for the year. So that's a lot of thinking about blue whale collective behavior. In the second section of the talk here, I'm going to talk about some other ways I've been thinking about integrating tools with collaborators to explore cryptic collective behavior in other elements of open and deep ocean ecosystems. So as we think about expanding our efforts to more species in the pelagic ocean more broadly, we have a couple of options currently in terms of thinking about behavior at this scale. One option, as I showed before, is to really rely on the fact that many of the animals inhabiting these ecosystems are producing a lot of sound, and we can use networks of hydrophones to study their behaviors over broader spatial scales. And this is something I've had the opportunity to do with a number of collaborators. For example, Emma Pearson, who's now a graduate student here at Oregon State. I was lucky enough to work with Emma and a team to leverage a broader network of hydrophones throughout the Northeast Pacific to understand spatial patterns of blue and fin whale vocal behavior. Also with an undergraduate researcher at Stanford, Natalie Cross, we've been expanding on some of this work to think about how marine heat waves can impact blue whale vocal and foraging behavior in the Central California current region that a lot of our previous work has been in. I think this is really valuable work for that really broad scale perspective on really the full ecosystem scale that these animals are interacting with. The other option that we can think about is really drilling down in more detailed nature in a specific location of not just recording the behavior of these predators, but also really in a detailed manner understanding the oceanography and the behavior and distribution of prey of these animals in one specific location. So this is work that's really been the focus of my time at the Monterey Bay Aquarium Research Institute over the last couple of years, where thanks to a grant from the Packard Foundation, myself and collaborators at MBARI have been deploying what we're calling our pelagic predator-prey behavioral observatory, which I will describe in some detail for you here. So just to set the stage of where we're operating again, we're looking at Monterey Bay again here. More specifically, we're zoomed in on this part of the shelf break where there's a shelf incising canyon branching off of the central Monterey Canyon. And this is the focal area for our observatory. And we've implemented a network of instruments focused on understanding predator behavior, prey behavior, and the physical fluctuations in the environment. So the tools that play a role here, I mentioned before these animal-borne biologgers in some detail in the earlier part of the talk. We also, to understand prey distribution and aggregation, we've been deploying an autonomous wave glider system that's carrying active acoustic instrumentation, so echo sounders, which are allowing us to map where prey are in the environment continuously in space and time. So in our observatory, this wave glider runs this circle that's shown in the bottom here, and it goes out for a couple of weeks at a time before we replace batteries, redeploy, and get a nearly continuous data record over the 4 months that we deploy this observatory for. In terms of thinking about other processes in the ecosystem, we rely on subsurface moorings here. These include hydrophones that we use to measure those predators' vocal and foraging behaviors, but also sensors like acoustic Doppler current profilers and CTDs that allow us to track really the fine-scale physical oceanography at this shelf break habitat. Finally, I'll mention, I mentioned before, MBARI's cabled seafloor observatory. One of the instruments that we're relying on a lot from that observatory in this work is actually a directional hydrophone or an acoustic vector sensor that not only records animal sounds, but points to where these sounds are originating from, providing a bearing to the animal's location. And that's been a really important part of this observatory, particularly as we've thought once again about blue whales and their interactions with krill. So I mentioned the seasonal element of upwelling before, how that can drive the seasonality of forage availability for predators, including blue whales. But previous work by my collaborators at MBARI, led by Kelly Benoit-Bird, showed that on much finer temporal scales, krill actually actively swarm on very fine temporal scales within upwelled plumes of water. And so this is an interesting dynamic to think about for a lunge-feeding predator like a blue whale that is selectively searching for these very densely aggregated prey to really try to understand, could they be tracking these upwelled plumes of water as a way to find prey in a really dynamic ecosystem. So just to spoil it off the top, in the study that we worked on with the acoustic vector sensor system, we found, in fact, blue whales do track these ephemeral upwelling plumes within the foraging season at fine spatial and temporal scales. So to show a little bit of what that looks like, here you can see where our directional hydrophone or acoustic vector sensor is located on the cabled observatory. We've got a really nice clear day with satellite imagery to show us this upwelled plume of water originating at Año Nuevo and flowing southward into Monterey Bay. Here's our directional hydrophone on the seafloor. And this is a bit of our cartoonish way to show what was really a data-intensive project here. But what we've shown is that under relaxation conditions between upwelling events, blue whales are typically hanging out to the northwest of our directional hydrophone, lingering off of Año Nuevo. But when these upwelling events occur and these plumes flow southward into Monterey Bay, we are able to use the directional hydrophone to track these animals around the compass rose and watch them follow this upwelled plume of water in which we know their krill prey tend to aggregate, at least in the Monterey Bay system, and really watch them track these ephemeral oceanographic features in real time. I think this is really exciting from the standpoint of understanding how these animals find food in such a dynamic ecosystem. But I'd also like to emphasize that I think there's a really important applied component here as we think about threats to blue whales in this ecosystem. So here are the locations of the main shipping lanes in the outer Monterey Bay National Marine Sanctuary. And what we're finding is that during this movement that occurs during upwelling events, these animals actually cross the shipping lanes and are likely exposed to greater risk of ship strike, which we know is a significant threat to this population. Another interesting element here is work led by my collaborator, Dave Cade, showing that in these upwelled plumes of water where you can get really dense aggregations of blue whales, what we sometimes refer to as supergroups of blue whales. You can see a nice drone shot here of a few of them, but we've seen up to 40 blue whales in an aggregation in these upwelled plumes of water. We've shown that when these formations of foraging blue whales are forming, in the hours leading up to those social foraging aggregations, we see an incredible density of a different blue whale vocalization, these type D calls. And that's prompted a lot of questions about what could be the function of these D calls in either mediating social interaction or providing information of the location of food or competitive dynamics between blue whales, and really prompts more questions than answers. But to start chipping away at those questions, I mentioned Kanoe's work before. She's now working with us as a master's student for her degree in computer science. And she's been helping us develop artificial intelligence tools to detect these clusters of decalls that we've previously associated with these social foraging aggregations. And some of the questions I'm really interested to get into using these detections of decalls alongside the rest of our pelagic predator-prey behavioral observatory is to think about the roles of physical forcing, collective behavior, and the interactions between these two dynamics in forming these ephemeral hotspots of trophic interaction. So to sort of break that down a little bit, that's a lot there. From the krill point of view, what drives swarm density? Is this purely physical forcing interacting with the bathymetry of the Monterey submarine canyon? Or are krill actively swarming as a behavioral response to that physical forcing to hold position as shearing forces increase in the water column? Or is it a response to the presence of predators in the ecosystem? From the blue whale point of view, thinking about collective behavior and collective foraging, we really want to understand what is the function of these acoustic signals and why are they producing them in such anomalous rates during the formation of these social foraging aggregations, and under what sort of biophysical conditions do these aggregations occur? This is work that, again, just starting to get into wrapping our heads around analyzing data to answer these questions. I'm really excited because we're now going to be integrating not only the empirical in situ data collection, but we're going to start integrating simulations of animal behavior again, like I mentioned in the work on migration before, thanks to a new grant that we just found out about a week ago from the National Science Foundation to build on this work. So to come back to our predator-prey behavioral observatory, I've talked a ton about blue whales and krill here, but the sensors that we're deploying here are not really species specific, right? This is passive acoustic monitoring, which detects the sounds of many animals, or active acoustic echo sounders, which detect aggregations not only of krill, but also of other species. So I'm really excited about using this system also in the present and in the longer term to really start to understand the behavior of some of these more deep diving echolocating predators as well. So as an example of that, we've started to explore behavioral patterns of sperm whales and their echolocation in the Monterey Bay area. Can enjoy another whale sound since I've been fire hosing information at you all here. So these are the echolocation clicks of a sperm whale, which I was surprised as I started to dig more and more into the Monterey Bay Aquarium Research Institute's long-term acoustic data record of how regularly these clicks seem to be in the background when we were listening in for blue whales. And that prompted a project that I led earlier in my postdoctoral work at the Monterey Bay Aquarium Research Institute, building automated detection tools to go through that long-term acoustic data record to identify when sperm whales are present and foraging in the Monterey Bay region. And to sort of situate this in a broader context, I think this could be valuable for insight into how sperm whales use space in the broader Northeast Pacific. So a variety of studies using sightings data, some telemetry data, genetic data, has indicated that sperm whales are moving across wide range of latitudes and longitudes in the Northeast Pacific. But in terms of these latitudinal movements, there seems to be a lot of murkiness still, understandably, given how cryptic these animals can be, in understanding the regularity of these movements, whether there's seasonality to them, and what the behavioral context of such wide-ranging movements might be. So to answer this question, we relied both on the Monterey Accelerated Research System, that's that cabled observatory, or MARS, that I described before, and also previously published data from the Gulf of Alaska, largely from researchers at Oregon State University, yet again. I won't go into the deep details of this project, but just to briefly break it down here, over many years of recording, what we're finding is that these animals are present year-round across these latitudes, but with subtle seasonality that's inverse across latitudes, which I found very interesting when we first plotted this for a couple of reasons. First is that talking to a lot of whale watch operators in the Monterey Bay region, people are not seeing sperm whales very often in this ecosystem, maybe a couple times every several years is the answer I've gotten talking to people who have eyes on the water a lot more than I do. But I think this points to the power of passive acoustic monitoring for understanding the behavior of these open ocean animals, because we've recorded now over 8+ years of recording sperm whales present and detectable from our hydrophone roughly 40% of days since we started recording. I also think this is really interesting because we have this sort of subtle seasonality at play here, which to me was a little bit unexpected for an animal that forages in the mesopelagic, where we don't think of the ecosystem as being particularly seasonal in nature. Again, I'm happy to take questions later if people have questions about elements of this, but yet again, we relied on simulations to try to parse these patterns a little bit here. So along with my collaborators, we used a wide variety of telemetry data from species across terrestrial, aquatic, marine systems, central place foragers, migratory animals, nomadic animals, and using distributions of their turn angles and step lengths associated with these different life history and movement strategies, we were able to develop simulations of what would it look like in the Northeast Pacific if these animals were nomadic, and what would it look like if they were central place foragers, and what would it look like if they were seasonal migrants. And what we're finding when we overlay artificial hydrophones in this simulation domain is that a seasonal resource tracking migration creates the best match to our empirical data. And by a resource tracking migration here, I mean seasonal latitudinal movement to track seasonal latitudinal variability in prey, and not a seasonal latitudinal migration like we were describing earlier for blue whales, where it's a relatively straight line migration between two distinct habitats, the so-called two worlds migration. So this is really a hypothesis more than anything here. What we're excited about is we're documenting the presence and seasonality of these animals in the Central California current ecosystem. But by using these simulations, we're able to hypothesize what might be underlying those patterns in space and time. And I'm excited, again, turning back to our behavioral observatory, to leverage this system to understand the finer scale interactions these animals are having with the prey and the environment, but also to guide efforts to tag these animals to really understand movement at the individual level in this ecosystem. I'll also just note very briefly here that this is work that we actually also just found out last week is getting funded by the Office of Naval Research to move towards tagging some of these deep diving predators to understand how they use this ecosystem in space and time, particularly in relation to the prey data we've been collecting from this observatory. Okay, so now that I've spent a lot of time talking about diving deeper into the ocean, in the few minutes I have remaining, I'm just going to end with a couple of quick notes about ways that I'm really excited to engage communities with this research and take it. We're talking about a lot of high-tech instrumentation, a lot of simulations and things like this, which I think are very interesting from a behavioral ecology theory standpoint, but really taking this into societally a relevant context here. So just as a reminder, we're operating this extremely dynamic space. An idea that I've been really interested in, and I know a number of people I've talked to here at Oregon State are interested in it as well, is this concept of dynamic management, where we really try to manage ecosystems that are moving in space and time with protection that moves in space and time as well. So here We're looking at what we might think of a classic protected area that stays in the same place in the same time. A dynamic protected area will be moving in space and in time in a dynamic management effort. And in a review paper I worked on a few years ago with collaborators thinking about dynamic management strategies, we did a survey of existing dynamic management programs in terrestrial and marine ecosystems and characterized the spatial scale and the temporal scale at which they update their management action. And what we found is really, I think, interesting in that there's some overlap here, but you'll notice in particular the marine systems require this updating on really fine temporal scales because the ecosystem is so dynamic on fine temporal scales. And as a result, we're really reliant on near real-time data, on forecasts, on algorithmic tools, basically, to have these sorts of dynamic management programs. And when we think about data and algorithms driving management decisions, I think there are a lot of societal ethical questions we need to be thinking about there. Some of you may have seen this paper that came out a few weeks ago in Science about biodiversity data globally and whether it really reflects biodiversity in animal distribution or if it more accurately reflects patterns of human behavior, right? Ultimately, biodiversity data is our observations of these ecosystems. And as we increasingly start to use this data in automated ways and algorithmic ways to drive policy, there's real questions there of who's represented by these data that we're relying on so heavily in our decision making. So in thinking about that, I think there's an important question to think about: how do we enhance the equity along every step of this process, right? So this is a paper that I was lucky enough to work on with collaborators mostly from the social science realm a few years ago that I learned a ton from in working on this paper, thinking about ways that you can try to enhance the equity of this process in every step of the process, from project development to how you design data collection processes to how you actually assess algorithmic solutions that you might be proposing for a management action. And we were thinking about this in particularly in the context of the High Seas Treaty, the agreement to protect 30% of the open ocean by 2030. But this is really something that applies not just in that context, even though I think that is a unique context because it's areas beyond any nation's, individual nation's jurisdiction, which creates interesting dynamics. This, of course, is also relevant as we think about the industrialization of our more coastal oceans and making management decisions there, and how we engage with communities in doing the data collection that's going to ultimately drive decision-making in this space. This is something that my collaborators and I have been thinking a lot about in terms of wind energy development off the central coast of California, deploying long-term acoustic monitoring stations to understand baselines of soundscape conditions and animal behavior, but also potentially changes as we see this industrialization. So this includes hydrophones I mentioned deployed with MBARI, the NOAA Sank Sound project, and then most recently in collaboration with the proposed Chumash Heritage National Marine Sanctuary and the Northern Chumash Tribal Council. And this is work that for me has been, again, a huge learning process and a really valuable opportunity to see how this work can be done in collaboration with a variety of community partners. I will say a huge thanks to my collaborator, Lindsay P.V. Reeves from NOAA in this work, who's really done an amazing job of pulling people together to not only think about these ideas, but also to go do this work together from NOAA vessels. And it's leading to what I think is some really exciting science. For example, my collaborator, Ella, who's a graduate student at Scripps, has been developing AI tools using this data to explore fish sounds and spatio-temporal patterns of fish mating behavior. And it's really inspired me to think even more about how do we actually bring this sort of recording technology to a greater number of people to enhance both the quality and quantity and equity of data collection acoustically. And this is something I've been lucky enough to be involved with a project from NASA where they've been using these really low-cost acoustic recorders called audio moths to understand how animals respond behaviorally to the recent solar eclipses we've been having in North America. And I'm really inspired by this program as a model for understanding other ecological and ecosystem questions, really by distributing data collection amongst people that really want to be engaged and are really excited about being a part of the scientific process. So something I'm really excited to move towards going forward. All right, so in closing, I've talked to you all long enough. I just want to end with a quote, not from me, but from someone much better spoken than me, the amazing acoustic ecologist and musician Bernie Krause, who I think said it better than I ever could in terms of the real value of acoustic monitoring for understanding animal behavior and ecosystem processes. And just a couple of things to highlight here is that he emphasized these are significant messages that we're recording for any sentient being. They're the sounds of survival, reproduction, and communication. If we can understand the behavioral context of animal sounds, we have a real opportunity to understand how they survive and thrive in these dynamic and changing ecosystems. I'll just emphasize that while I've been thinking about that, mostly how we try to interpret individual and population level acoustic data in marine systems, we can also do this in terrestrial systems. And as we zoom back out to what I mentioned about my interest in animal behavior in dynamic and changing oceanic ecosystems, hopefully we can start to integrate tools to understand not just oceanic ecosystems in this way, but understand these processes across Earth's ecosystems, which is really would be me feeling like my grand ambitions in my career were fulfilled. Maybe we'll get there like decades from now. Anyway, this is obviously work that requires a huge number of collaborators who are incredible people to work with. I mean, I hope that came across throughout the talk, but I really benefit from working with and learning from an incredible diversity of researchers and institutions. And huge thanks to the funding sources and the incredible artists that provided some illustrations for this talk. With that, I'm happy to take questions. Thank you all for listening.
Thank you so very much, Will. That was a lot. So I'm going to guess that there's some questions out there, but I'm going to start to see, is there any questions online? Okay, for folks that are joining us online, go ahead and enter any of those questions in the chat box and we'll work through them. How about questions in the room? Okay, I'm going to start in the back, so you're going to have to give me a second. They do it on purpose, I swear.
Hi, that was an amazing talk, and I'm so grateful to hear it. And I was wondering, because you did mention that we could talk about this in the questions, do you mind going more into your metrics for understanding the upwelling, the seasonal upwelling?
Yeah, for sure. That's a important question. Let me find a slide here for you. While it's loading, I'll just mention that the metrics that we, as a recap, the metrics that we looked at in that work are both the timing and intensity of upwelling. So there's a couple of different metrics we can use there. In particular, and this is work that I'm pulling from previous work done by physical and biological oceanographers, a paper led by Stephen Bograd a number of years back from NOAA, showed methods of identifying when you're getting the spring transition, when you're getting the peak in upwelling intensity, and when you're getting the cessation of upwelling accumulation. And that's what's shown here in that onset, peak, and cessation. And then you can look at that total cumulative sum, as well as the cumulative sum between each of those time points in the seasonal cycle. And so that provides us a real diversity of timing and intensity metrics, and metrics that integrate timing and intensity, which we're able to explore statistically, really simply through linear regression, like year by year, when are animals at the population level migrating south, and what did the onset timing look like? What did the peak timing look like? What did the cessation timing look like? And ultimately what we found was significant relationships, again, with earlier onset, a later peak, and a greater total accumulation. And as you can see from these figures, those things tend to be correlated amongst themselves as well. So it's really almost like typologies of upwelling seasons, right? So a year like 2020 in yellow here, we had a very early onset, we had a very late peak, and we saw a really high amount of total accumulation. I'll just note we published this study a few years ago. Since then, we've seen even greater variability in upwelling conditions in Monterey Bay, really topping out the scales here on terms of the long-term climatological patterns that are overlaid there in gray. I hope that answers your question. Okay, yeah, maybe we can chat a little bit more afterwards.
Perfect. Okay, I saw another question in the room.
Yeah, I wondered about this seasonal variation and which means that the females are going south, say, later in a year. How does that affect their mating and then their calving? And since they're coming back about the same time each year, that means they're coming back with younger calves, for instance. Or sorry, the following year they will be. I mean, has anybody looked at this?
That's a really interesting question. I would love to dig into that. I don't have a really good answer for you. One thing I will say is that you may have noticed that the timing of that transition from foraging to southward migration was really variable year to year. But the presence of song in the ecosystem, regardless of if it was coming from foraging or migratory animals, was actually quite consistent year to year. And so what I think that's indicating to me is that we're measuring the migration turnover or transition specifically at Monterey Bay. Whereas we continue in some years where they migrate much earlier, we continue to hear animals migrating past our hydrophone for weeks to months after that local transition. And so really what this points to is the work that I mentioned in collaboration with Emma about needing to do this work across a latitudinal gradient to understand, are those transition timings consistent across latitudes? Because I think that's really relevant to your question when we think about like timing of arrival at breeding grounds or calving grounds, the timing of reproduction and things like this. It's not something that we're able to answer from one location. But yeah, it's a really great question.
Thanks.
Other questions in the room? Oh, there's some questions.
That was really awesome, Will. I am just going to ask, and I know this is probably not your shtick, but you know, there are a lot of organisms in the ocean that don't communicate with sound.
Yeah.
So it seems like you have like this generalizable framework, but I guess I was wondering, how generalizable is it? And it seems like there's a lot of really cool like acoustic monitoring, but I'm going to speak for the sharks here, which will probably surprise nobody. Like when we think about acoustic monitoring, it's different.
Yeah.
And so I was wondering if there were parallel efforts to look at how other predators are moving in response to these environmental variables and in these areas that you're also monitoring with different types of acoustic technology.
Yeah, it's a great question. This is like a really important point. We're monitoring animals that make sound, and even within species that make sound, we're monitoring the individuals within those populations that are making sound. Also an important caveat. In terms of animals that are not producing sound that we're able to detect from these hydrophones at all, I mean, gosh, I want to like ask you this question basically in terms of acoustic telemetry, for example. Like I do know of work, and I'm sure you know more about this work than I do of using acoustic receivers and acoustic tags implanted in animals to understand movement across latitudinal gradients, also social dynamics, which, yeah, it's tricky whenever, I think one big advantage we have with the sound producing organisms is it's not only an indication that they're present, but it's an indication of what they're doing, if you understand the behavioral context of the sound. And it's also, they are producing a social signal as well, right? So there's all of these layers of useful information for social behavior questions. Whereas as I understand it from acoustic telemetry, where you're receiving at a hydrophone that's listening for acoustic tags, you can gain some information about who's there, but in terms of like interactions, I think there's a lot of questions there. One thing I've wondered about in that space is stuff that I have seen from the terrestrial realm where people use what I've heard referred to as like proximity tags, where animals are like interacting. I've seen a lot of work on that with bats, for example. I'd be curious to know if that's something that you all are working on. Maybe we can have like a deeper conversation
We should chat.
later.
Yes, the short answer is yes. That's what we're doing with the basking sharks in Ireland. So
Oh, super cool.
we can talk about that. But
Okay, wow.
yeah, in general, it's interesting to think about the challenges with studying and the fraction of animals that these types of technologies might not be capturing.
Oh, for sure. Yeah, and I think that's a huge part of the interest in that predator-prey observatory system I was talking about, is it allows us to tap into players in these interactions that are not going to be detected on the hydrophones, for example, the technologies I listed there still don't cover the full suite of organisms at play here by any means, and that's a really important point. Yeah.
All right. We're going to take one more in the room question and then we'll wrap up.
Hi, great talk, by the way. That was fantastic. I know you spoke about more cryptic animals like the sperm whales, but going even more cryptic, do you think the acoustic listening would be applicable to listening to beaked whales or something along those lines?
Yeah, that's a great question. I'm so glad you asked about beaked whales. Yeah, there's a couple of thoughts there. This is one of the things I really love about the passive acoustic monitoring is like, for, again, an animal that's going to produce sound, there's so much to work on here that it's just promotes collaboration in my mind. So we've recently started collaborating with a graduate student at UC Santa Cruz, Allison Payne, who is using the long-term time series to try to study beaked whale presence and seasonal patterns, interannual patterns, things like this. The other thing that I'll note is that I'm really excited about acoustic recording from animal-borne devices for monitoring things like beaked whales. And this is, I'm totally stealing this from Allison. She's been working on deploying tags on elephant seals carrying acoustic recorders out into the Central Pacific as a means to monitor or even see if beaked whales are detectable in that ecosystem, given that they're foraging in the same ecosystem very cryptically. Yeah, beaked whales, I mean, they're kind of like, I think a really good poster child for like the power of passive acoustic monitoring, because like, how else are you going to do this? Yeah, but they're also really challenging, even within the acoustic space, given the really high frequency nature of the sounds they produce. But yeah, that is something that we're starting to work on and haven't gotten deep into yet, but yeah.
So I think that was an invitation for collaboration. And with that in mind, I'm going to hand the mic back to Dawn for a second.
Thanks, Will. That was awesome. I meant to say this earlier, but we are going to head to Beer One afterwards pretty soon. So if you want to continue chatting about science with Will or hang out with friendly people or feel like you need a beer, join us at Beer One, 5-ish or whenever you want to migrate over there.
All right, let's give it up for one more time for our speaker today. Thank you so much, Will. And for everybody in the room, I hope to see you again next week at our student invited speaker. Thank you, everybody.